Okay, today on the podcast, we are honored to have Emma Lewis and Morgan McCrea, first year students in the UTSA Developmental and Regenerative Sciences PhD program. They're here to shed some light on the research being done right now in Alzheimer's world. Hi, Emma. Hi, Morgan. Welcome to the
Hi.
show.
Hi, thank
Hi, thanks
you.
so much for having us. So excited to be
We're
here.
We are so excited and honored to have you. So, to just jump right into it, can you give us a brief overview of your research and what you're studying in your PhD program?
Yeah, for sure. I can start us off. The PhD program, as you said, is the Developmental and Regenerative Sciences program, but it really encompasses a lot more than just that. It's very similar to what students classically study is more like cell and molecular biology. And then specifically in our program, it's within the umbrella of like the neuroscience, developmental, and regenerative biology department. So there's actually a ton of neuroscience in our program, as well as like a big group of scientists in our program with the Brain Health Consortium. So there's a ton of different kinds of things that in general we study in our program, like disease, fundamental biology, neuroscience, etc.
Yeah, and then a lot of our department also does stem cell research, looking at male fertility, different organoid models for brain development and brain diseases. So that's all really interesting stuff, and it's a really cool overlap of different research topics under one big umbrella department of neuroscience, developmental, and regenerative biology, which traditionally seem kind of different topics in the scientific world, but it's nice to be able to combine them a bit more translationally.
Yeah.
Wow. Wow. Okay.
Yeah,
It's really impressive.
Thank you. We're still in our first year, so that the first year of our PhD program looks a little bit different from what the rest of our research trajectory will look like, since we're still so much in the learning and training phase. So for our first year, you have three laboratory rotations, almost like kind of semesters, but for your lab work instead of classes. So you get to have a different experience with different principal investigators, other students, and different techniques and areas of research that they're interested in. So it's a really nice way to get a widespread first experience when you might not know exactly, you don't even know what you don't know yet. And so it's kind of nice to have some experience like that.
for sure.
That's great. How did you become interested in neuroscience and specifically in developmental and regenerative biology?
So for me, I became interested in neuroscience, honestly, for personal reasons. When I was a teenager, one of my grandmothers was diagnosed with ALS, and very shortly after, my other grandmother was diagnosed with Alzheimer's. And they both lived about 1 mile from me, and I was used to seeing them every single day. And before that, they were perfectly healthy, full of life, and just to see them deteriorate so quickly and in completely almost opposite manners was obviously shocking and hard, as anyone who knows anyone with any sort of neurodegenerative disease can understand. So that just really triggered all of these questions for me, and I just couldn't wrap my mind around how this could happen and how it could happen so drastically, so quickly, and how many people are affected. And so I just could never really stop thinking about it. So that was for me and I was always fairly curious, but that was what really set me off on this path.
Yeah, for me, it's been a little different. I've already told you guys, Morgan and I are both here because she does very specifically neurodegenerative disease research and is interested in Alzheimer's disease. For me, I started off interested in Alzheimer's disease because also for personal reasons, I wanted to learn more about it because both of my grandmothers have also passed away from Alzheimer's disease, and that's something we've been dealing with like this year and last year a lot. It's very new for me. But then I got more interested in more fundamental biology and development, just definitely more of the developmental and regenerative sciences portion of things. But it's really, really cool to see how it all connects and to be in this community with everyone. Even right now, like I think what Stephen mentioned to you, like in my lab I'm working in now, is working a little bit with Alzheimer's disease patient cells. So, it's always relevant, and it's a big part of my life. So.
And when you say Alzheimer's patient cells, you mean like people that are still alive?
Um,
Is that true?
I am actually not sure. I think it's probably a little bit of both because you can culture, it's essentially stem cells with human-induced pluripotent stem cells. So you can take them from blood samples. I think that's what typically blood or skin. But if you have blood samples from Alzheimer's disease patients, their protocols, I'm not totally familiar with how it's done in the lab, but you can induce them into like stem cells you can culture forever and ever and work with those. We'll get into it more, but there's a lot you can do with the stem cell technology that we have available.
It's fascinating.
Yeah, it's very cool.
Yeah, I think I was telling him like it feels a little surreal sometimes to just have like Alzheimer's disease patient cells in the palm of my hand. It's like, this is real stuff.
And when I was younger and my grandmothers were sick, they both participated in clinical trials. And I didn't really know that at the time or necessarily know what that meant, really. But as I got more involved in wanting to become a scientist myself, my parents told me, and they were like, hey, your grandmothers both participated in some of the stuff that I think that you're wanting to do yourself. So that made it even more special to me to know that they participated in this kind of thing as well. And I don't know, there weren't great records kept of my parents didn't keep great records of the trials that they participated in, so I don't know what they were. So I don't know more details than that, but just knowing in general that they were involved is really special to me and makes my own personal research matter a little more,
Absolutely. What do you think, Sue?
I think this is incredible.
I know.
I think you guys are amazing, and
Thanks.
I'm really blown away by the two of you. We often find when we interview people that they are, we haven't met anyone like you two yet, researchers and scientists, but even lawyers, speech therapists that we've talked to, they're helping out the senior citizens and the Alzheimer's patients because they were personally affected. And, you guys going through that too, and that hardship, but now look at you guys. I mean, it's really incredible. Really, you should be really proud of yourself.
Thank you very much.
Yeah. Gosh, I don't even know what to ask. These questions are crazy, Joe.
Well, okay, let's just, for those of us who are not in the science world, even though I did teach science for 15 years, can you explain what developmental and regenerative biology means and how it relates to the brain?
Okay. Yeah. I think I can do this one.
We'll tag team.
I think in this case, with researching developmental and regenerative sciences in the brain, when we say that, we're wanting to understand how like a single cell embryo becomes a multicellular organism, when you're developing like in utero from like conception, and you have all of these stem cells that can become all kinds of different cell types. And, it's important to understand how a cell decides to become a brain cell, and there's a lot that goes into that. And then additionally, in development, there's studies, it's important to understand how neurodevelopmental disorders come about, cognitive disorders during, after birth. And then that really ties into regenerative biology because being able to understand because again, we work with a lot of stem cells, which just are cells that can become all kinds of different kinds of tissue and cells. Sky's kind of the limit. If you understand how to generate a certain type of neuron from a stem cell, you can actually recapitulate that in a dish. And that's how we have neurons in culture. And then with that, you can study, regenerative medicine. Again, like, it's a ways off, but the
We're getting
idea,
there.
if you can, generate these new neurons from stem cells, you can come up with, transplantation therapies and stuff like that. You can study, manipulate them to study disease and study healthy cells. Like, it's just, I don't know. It's a big question. It's a world of science you can do there. And both of us are interested really in, like, and we'll talk about this more, like, causal, like, I guess, like, more fundamental, like, where the disease comes from, how, you have we're talking about, like, prevention.
Yeah.
That's what we're really interested in as well. I mean, obviously, both of her parents had Alzheimer's and dementia, or your dad had dementia. My mom currently has Alzheimer's. She's in a memory care facility here in the area. So yeah, we're really interested in what causes it. We're trying to like prevent it by doing all the things. So we really, like, you guys are really, like, I know it seems like you're just researchers or whatever, but you actually are really changing lives. Like, it's incredible.
Means so much. We're
One day at a time.
one day. Thank you. Means a lot,
Science
but
is slow.
it's a process. But I was really excited to start with this PhD program specifically because of the regenerative biology aspect. Since my interests are specifically degenerative diseases, I wanted to look at the flip side. And so we're able to regenerate some cells, some tissues. What could we possibly do to apply the features of those cells to cell types that might need a little extra help? Because neurons also, in general, they don't regenerate like a lot of other cell types do. Like, you get new skin cells all the time, you don't get new neurons. And so even a healthy neuron doesn't normally regenerate the same well other cell types would. So I find it interesting to study the potential factors with other cell types that make them regenerate. So potentially we could apply them to healthy neurons, and then in the long run, even the unhealthy neurons. And so I was really interested to come look at the neurodegenerative diseases from a regenerative perspective. So that's where I kind of came into this exact field. And it was really cool to find a program that really specifically so overlapped the regenerative biology aspect with neuroscience specifically. Yeah. So I was really excited about that.
It's great. Your school's doing really great things, which begs the question, how is your research funded? And is your funding affected by the recent efficiency protocols of our administration?
Yes, it is. Immediate answer is it's looking that way. A lot of our funding comes from the NIH, and with indirect research costs being cut, those are the costs that keep the lights on the buildings, routine maintenance that's required for super expensive and elaborate lab equipment. And so even though these cuts are in quote unquote indirect and not necessarily, they're not actually cutting the Alzheimer's disease research, but by cutting the funding to the building where you do the Alzheimer's disease research, it makes it a lot harder to do that. So we're hoping that with all of these judge orders to halt the freezing of everything, hopefully damage can be kept at a minimum. But absolutely, even research that is not considered DEI or any of the other flagged, quote, whatever they're calling the flagged research, even that research, that's not all that's being affected. Everything's being affected. Alzheimer's disease, biomedical research, genetic diseases, everything is being affected. Yeah.
And
exactly.
we're in an okay position right now as first year students in the program funded by the department. It'll be a problem in the future, and it's really a big problem right now. they're rescinding offers for incoming first-year students left and right.
And
like,
faculty hiring is more difficult.
Incoming students are
funds are unsure,
left with nothing. Yeah, because they're like significantly cutting the number of students that are being admitted into these programs. Yeah. So like, I mean, there's an immediate consequences of like just the amount of research going on being reduced because of it's impacting trainees, and we could talk about like international trainees as well. That's a whole thing.
There's a whole it's a whole rabbit hole.
Yeah, the supplemental funding, so like diversity funds for like undergraduates and trainees in the labs are just being cut out of grants because you can have like extra money tacked onto a grant for other just like workforce hires, undergraduates and stuff that are just getting like cut off. It's just, yeah.
It's a mess.
We're at the mercy of.
We're being told it's an indirect cut. Alzheimer's research is not being cut, but it is effectively, right? Is that what you're telling us?
It affects everybody.
It's being affected. What they're saying is technically true. No, they're not cutting the Alzheimer's disease research, but they are cutting all of the indirect costs, therefore hindering the ability to conduct Alzheimer's research. So essentially, yes, it's being affected for sure, 100%.
Yeah, it's just I'm not super informed on like all of the legal aspects of everything. Technically, none of it is legal.
And that's what I'm hoping that the judge, I'm seeing more orders for them to have to halt what they're doing. So I'm just hoping that they keep the damage at a minimum because so much of it is illegal. So I hope that I'm choosing to be optimistic.
Yeah. I mean, what can we do?
Yeah.
Love it. I mean, yeah. What else can we do? Okay, so let's get into like the crux of Alzheimer's. Are you studying how to prevent it, how to slow it down, or maybe even reverse some of the damage? Like, what do you know about that?
So
or what can you speak?
We're kind of doing different things right now. So that's honestly great because we kind of cover a couple bases. So I'm wanting to look at some of the causation aspects and therefore possible preventative measures as well. So that's something that I've found really interesting. Again, I'm still kind of just getting my feet wet with my own research, but it's really been exciting to start doing that. And I can talk a little bit more about the details of that in a second, but Emma's doing a different avenue, so I'll let her give a little intro with that.
Sure. Yeah, there's a couple things. What I specifically am working on is more like a technical like methodology for silencing genes that are more highly expressed in Alzheimer's patients.
And what's that mean exactly? Can you?
Yeah. It's just in disease there are often, I guess, I don't know, that might be the best I can do. Like there are just certain aspects and traits that like really stand out in these diseases, and there's ways to like basically silence them and then see what happens. And those are just very like molecular ways that you can identify like a disease versus a healthy cell.
Like the phenotypes.
Yeah. Well, phenotypes. Yeah.
I'm asking.
Yeah. Yes.
Okay.
So it's more like.
Wait, those of us that are not in the know, explain what a phenotype is.
See, that's what I'm saying. Like now I have to explain what a phenotype.
Well, yeah, but we have to. I mean,
I was trying not to say phenotype.
Isn't very.
Well, there's like your genes, and then the way that they express, like your hair color, your eye color is your phenotype. It's what you can see, like what is the physical, like the way it's, okay. So things like that. Like what I'm really doing is more like a technical, like CRISPR editing and stuff, but that's like a whole nother avenue. There's another project going on with a neuroscience PhD student in my lab, which is super interesting. It's more of the stem cell stuff, except skipping the stem cell step. And how can I explain that? Like he's trying to make like human, like wound healing cells, like directly into a type of neuron. Skipping, you can go from a like skin cell to a stem cell to a neuron, but when you go to the stem cell intermediate, it like wipes out all of these essential aspects of the cell, And so he's trying
essential
to skip
for its
that.
later development.
Essential markers of aging, actually. It kind of like renews the cell to like its youth, and if you can skip that step, you can have neurons where you can actually study aging markers. And so he's working hard on that, and it's more of like a modeling situation where like there's a lot of research going on here where people are trying to model disease in a dish or have a good representation like that and then study it later. So that's kind of like what I'm doing.
And the modeling in the dish can just give an easier environment to manipulate and study exactly what the cellular and molecular aspects of the disease are doing that aren't easy to do in a body. So
yeah,
just gives multiple avenues.
And then Morgan, you're doing something a little different, correct?
Yes. So I am wanting to study what's causing Alzheimer's disease. And so what I'm looking at is a metabolic and energy component. And so the kind of hypothesis idea that I'm pursuing is that there is a breakdown in the metabolic pathways of some sort that basically damage the powerhouse, the mitochondria of the cell, and then that causes all sorts of problems. And that's what I think that's a good avenue of study, especially based on current research that I've seen over the past few years, looking at the importance of respiratory health and cardio in Alzheimer's prevention, and they're finding it to be so linked to brain inflammation. It's been really interesting to pursue a different aspect than what has previously been looked at, because that's something that the field has realized in the past few years. We're starting to look into some other avenues of research than what has historically been mostly focused on. So there are a lot of new ideas that have been popping up, which have been really exciting to start investigating.
Do you feel like your research will result in any sort of treatments for Alzheimer's, say, in me and Sue's lifetime?
It's hard to say. It's hard to say because things can happen so slowly or so quickly. It really depends on what we find. If something is found to be very correlated, then it's really exciting, and that's an area that's going to be a lot more quickly pursued. But if we find less results, it's going to take a little longer to keep working. So I think it really varies, and I think the next few years will be very telling to see where, especially the regenerative medicine field goes, because it's still a fairly new field in science, and there's a lot of ethical ramifications they're working through still. So we'll see. I could see it going either way. We'll see. I hope so.
Yeah.
I hope so too. And my follow-up was going to be, do you think that will involve stem cells?
I think a lot of future medicine
in one way
will.
Or another.
Yeah.
In one way or another, because there's a lot of things you can do. Like we've actually recently, we learned about this in class.
Yeah.
You can take somebody's, there's different avenues. So not just like what you think of, I think when people think of stem cells, they think of like taking damaged cells in someone's body and like growing them back, or like making a new arm, or, stuff like that. But you can, kind of like how I explained, you can take like a sample of someone's blood and that has their, that individual's genome, like their, genes are in those cells and their serum. And what?
And why blood? Why can we take blood?
Because the blood cells can be turned into stem cells.
Because, their lineage that they're a part of is very adaptable and
malleable.
Malleable, that's a better word.
Yeah.
So blood's an easy, it's easy to extract, and it's a quick process to spin their blood cells. So it's a good option for.
It's good for, what is it? Like customized treatment. Because if you have those cells, you can, take someone's blood cells, make them into stem cells, and then make them into like, what is like your favorite cell type, muscle cells or something. and then you can test drugs on them. You can understand like how the muscle develops with like the whatever genes are going wrong in their cells. There's just a lot of things you can do. There's like transplantation therapy as well, but that is, I think, the furthest off. That is because it's just hard to. The problem there is that the cells, you need to have them know where to go. It's really that simple.
They basically need a GPS, and we haven't really figured that out yet.
Yeah.
So yeah, there are a lot of, and then there's also different types of genome editing that's also a little bit far off just because of ethical ramifications, because how do they know this gene really does only have one effect? So that's something else that I could see taking a while to get approved just because of the potential for off-target effects.
Yeah, right now I think the only CRISPR therapy available is for sickle cell anemia.
For sickle cell anemia, and then with patients, they still can't do anything on babies since they don't have a choice. So
that's fair. It's tough.
It's tough. So I think the field is advancing, but since this is all just so new, I think that there are a lot of things that have to be worked through to make sure everything's fully safe. But I'm hopeful.
It's super exciting.
It's exciting to see the progress of science in general, year to year, 5 years, 10 years. So whenever I get a little frustrated, I take a step back and look at the bigger picture. And I think about what we didn't know 10 years ago, so it gives me hope. So I think it's exciting to see the field's progressing.
I love it. And that brings me to my last question. Where do you guys see yourselves in 10 years?
Ooh,
for me,
that's exciting.
10 years. Yeah. I am at a place right now where I still want to do research forever.
Me too.
So, PhD hopefully will take like 4 years. Yeah, 4 years. They sometimes will take a lot longer than that. And we're like, that's
4 to 7
ridiculous.
years is
7 years is too long.
Yeah, I want to be done sooner than that too.
I want to keep doing research. I want to do my PhD and then do a postdoc, which could be, people do postdoc research, like postdoctoral research. You just join a lab, do whatever you want. Basically, you could just get funded. Hopefully, the funding situation is improved by then.
Yeah.
And you can do that kind of research anywhere from like 2 to 7 years. So, I mean, in 10 years. My overall goal is to be a principal investigator myself, get a faculty position, and run my own lab. The thing is, I just don't know exactly what direction I want to take yet. I've actually, since joining this program, I'm like notorious for changing my mind. As like,
yes,
I'm like, I was really interested in neurodegenerative diseases, and now I'm really interested in like reproductive and fertility and development. So, I mean, it could all come together someday. Maybe I'll study neurodevelopment. Who knows? I just follow like whatever like interests me, like from day to day. Suddenly, that's my favorite thing. Hopefully, that can work that to my advantage. And
leaves you with some open paths. I personally, I don't quite know exactly what my future will look like, but I also, like Emma, know for sure I still want to be doing research. I want to be a neuroscientist for life. But I do want that to change a little bit as I grow, which is one of the reasons I wanted to do a PhD fairly early in my career. I wanted to have the opportunity to develop as far as I see fit based on the experiences that I have. I would love to go into some sort of industry job for a little while to get, I don't know, I think drug development would be really interesting, something I would really like to do, but I would also love to teach at some point. So I don't really know what order that would come in, but I know I want to do both at some point. So I'm just keeping my options open, and I'm trying my best to look at what's out there and also know that things might open up in the next few years. And like Emma said, it will depend on the funding situation, what that all looks like.
Everyone who wants to do research needs to have a plan B.
Yes.
It's just everyone has a plan B.
Especially
even
right
faculty
now.
members have plan Bs still. Like tenured faculty members still have a plan B.
That is actually so sad.
I would agree.
I think it really has more to do with like how much work it is. Like it can be a very painful job to have. You have to be very like dedicated.
Yes, but I also think that it's I think there are a lot of things.
I
Well,
have to
listen,
be steered away yet.
we just thank you so much for coming on, educating us, for all the work that you are doing to really change lives. And Sue and I were just talking about how we just love your generation so much. You guys are learners,
Thank
you're
you.
leaders.
We're
That
just
means
doing,
so
we're
much.
just
It
doing our
means a
best
lot.
out here. Like we're just surviving.
Thank you.
Thank you.
It really means a lot.
It's been great. Yes.
I have a question. How did you find this program? Was is there a lot throughout the country that, something that you really specifically wanted to focus on? Like, how did you end up at this school and this program?
So for me, I ended up at this school, at this program very, very specifically. During one of my college classes, we had a research assignment, and I picked Alzheimer's disease since that was my interest. And I found this scientist who produced a lot of papers that were so interesting to me and a different avenue of Alzheimer's research than I had previously been super familiar. And it was focused on mitochondrial dysfunction. And it blew my mind, changed my world, and I was just so fascinated and was like, I have to work with this scientist. And I found out he was in San Antonio. And coming from UT Austin, I was like, well, that's really convenient. That's not far at all. So that's how I ended up here. I wanted to work with one guy specifically, and here I am. So very lucky.
Awesome.
Very lucky.
See, that's a great story. I don't have one of those. For me, it was definitely like looser. Like I was looking all over the place, but also like local in Texas. I did my undergrad at Texas State University, and I just knew like my undergrad research was more like it was a neuroblastoma like cancer treatment research. So with like that experience, I just knew I wanted to do some sort of like cell bio like disease research avenue. And I honestly just thought like the DRS program seemed like really cool and catchy, and like the people seemed nice, and so I applied. And like I had a really fun time like meeting all the faculty and interviewing and stuff, and I was like, yeah, that's the place I want to be.
Mm-hmm.
And like I appreciate still like there's a lot of different research going on in this program, and that has benefited me greatly because I've rotated in like 3 very different labs now. I've changed, like I've had a lot of different interests already even within the first 2 semesters I've been here. So it was good. Like something I definitely was looking for was a program that was pretty broad with like the areas of research available.
Mm-hmm.
And there's a lot of collaboration too, which is just really awesome.
Yeah. It's really nice.
Mm-hmm.
That's
Love
amazing.
it.
Sue, you have anything else?
No, I don't.
I don't either. I just so happy y'all came on to
Yeah.
talk to us.
Thank you so much.
It's been super fun. Thank you.
You're welcome. And we want to keep up with you and see like how things are going.
I love
So
that.
sorry if we become like stalkers, but
No, I'd love it. I'd love it.
We want to come to the lab.
Yes, we would love
Yes,
a field
sure.
trip. Field trip. It would be so fun. It would be really fun. Well, and I really appreciate that you guys do this podcast and bring light to something that affects so many of us and is so personal and hard to talk about. So thank you guys for doing this podcast, and thank you for your interest in baby researchers like us. So really, really, really appreciate it. We were looking forward to this a lot, and you guys are so awesome. So
Yes.
thank you for
Super
everything.
awesome. Thank you.
We've been looking forward to this. This was I think it's our favorite interview.
Oh my gosh, you guys, it's too sweet. It's been like my only interview.
I know, it's been so amazing. I know, I love it. I love it.
all right,
you guys
well,
Podcast.
thank you guys so much, and we'll talk to you soon.
Yes, would
Yes,
love it.
thank
Thank you
you.
so much. Happy Friday.
Happy
Happy
Friday.
Friday. Okay, I'm gonna hit
Okay.
end,
Okay.
and then.